From medical breakthroughs to breaking health news, from one of the great medical capitals of the world, this is NECN Health Week.
Up next on Health Week, a researcher shares his excitement over a groundbreaking stem cell discovery that could lead to a cure to osteoporosis. Osteoporosis is the leading cause of disability among older adults. In fact, more than 500 million people have it. Now, a breakthrough announced this week in Boston leaves researchers optimistic that they may have found the eventual cure. Here to explain it all in layman's terms is Dr. Robert Sackstein of the Biomedical Research Institute at Brigham and Women's Hospital in Boston. What an exciting time to be in this research field.
Well, it certainly is. And the most exciting news about it is that we could potentially be harnessing the body's own repair system optimally with this technology.
Within perhaps my lifetime, your lifetime?
Well, we sure hope that it will be within the next decade. I think that there's a good possibility that this opportunity opens up the first clinical application of adult stem cell therapeutics for a non-blood cell disease type.
Okay, now this is what you've helped to repair bones,
Exactly.
Bone matter. Okay, how does it work in layman's terms?
Well, it's a very interesting biology. The bottom line is that the cause of osteoporosis is an imbalance between destruction of the bone and production of the bone. And as we age, the cells that destroy bone basically continue destroying at their own pace, maybe slightly faster, but not much. But the ones that make the bone, the osteoblasts, they start slowing down for reasons we're not clear. Some of it's related to hormones, to be sure, because estrogen and testosterone, the female and the male hormones respectively, both improve osteoblast synthesis of bone matrix. But beyond that, we're not quite sure why there's a drop-off in osteoblasts in the bone. However, the cells that make the osteoblast, the stem cell that creates the osteoblast, is the mesenchymal stem cell. And what we sought to do with this work was to get a platform technology that would allow us to deliver the mesenchymal stem cell to the bone, so we can make new bone.
I'm just going to draw an analogy that I believe I read in one of your papers, which is that it's almost like a GPS system. You can direct this thing right to the targeted area.
Absolutely. So what we did actually is we studied intensively the cells of the body that use molecules to the best advantage to move them around the body, and that's the white blood cell. So a white blood cell in blood flow will get to a site of inflammation or infection rapidly. And if it didn't do it with some precision and efficiency, we would die. So we sought to identify the molecules that a white blood cell possesses that allows it to get to those sites with high efficiency. And we asked the question, what is it that a stem cell is missing? Why wouldn't a stem cell be able to get to those sites as readily as a white cell? And we systematically went through that and identified a set of molecules in particular that drive the stem cell to migrate to the bone.
Okay, so you have been met with initial success. How far away are we from a human trial?
I think a human trial is imminent, within a few years to be certain, and maybe even earlier than that, depending on a few hurdles that have to be crossed with FDA regulations. But what we did is our work was with human mesenchymal stem cell, the precursor of the osteoblast, and we used a mouse model solely to prove the principle that we could drive the mesenchymal stem cells into the bone.
Okay, now if you, looking beyond its purpose as it pertains to osteoporosis, could it one day the same methodology be used to help fight something that might pertain to tissue or other matter beyond the bone?
Precisely. So as it turns out, the molecule that we were creating on the surface of the stem cell, which is done with a chemical engineering of the surface of the cell, so we didn't introduce any genes or gene products into the cell. We basically modified the cell surface and maintained viability of the cell in doing that, and also its capacity to differentiate or develop into osteoblasts. That modification, that molecule that we place on the surface, is the master molecule that moves white cells to any site of tissue injury.
So this is huge.
So it's beyond,
the potential is
tremendous.
Is
beyond what would be its most important immediate application, which is bone disease.
This is fabulous. We have just a minute left. Let me just ask you, are you as excited as the general public and surprised by the number of breakthroughs we're seeing all of a sudden, it seems?
No, I think there's a lot of work that's been done by many people that preceded our work, and certainly the accumulated effort of all the investigators in this field is coming focused on the use of this technology most appropriately, and I think we're heading down that path very quickly.
It is fabulous. Dr. Robert Sackstein, we wish you nothing but continued success.
Thank you very much, and I hope so for our patients. Thank you.
And I hope you can come back and tell us one of their success stories.
That would be wonderful.
That'd be terrific.